Great. So starting with number 79, the question asks, all of the following techniques are used for immobilized plant cell cultures except. All of the following techniques are used for immobilized plant cell cultures except. So which technique is not used for immobilized plant cell cultures? A, we have entrapping in an inert substratum. B, absorbing onto an inert substratum. C, covalent bonding onto a biological macromolecule. Or D, absorbing through biological macromolecules. So which one is not a technique used for immobilized plant cell culture? Or rather, what are the techniques used for plant cell cultures? If anyone knows the techniques used for plant cell cultures. No answer, we'll skip that. Moving on to number 80. The question asks, which of the following pharmaceutical products is not commonly produced using biotechnology? Which one of the following pharmaceutical product is not commonly produced using biotechnology? Analgesic agents, clotting factors, fertility drugs, or insulin? So which of the listed products is not produced using biotechnology? Stanley says analgesic agents. Yeah, analgesic agents. So clotting factors, fertility drugs, insulin, they all produced using biotechnology. If someone can mention a fertility drug that is produced using biotechnology, what is an example of one? Clotting factors, insulin, those are straightforward. What's an example of a fertility drug produced using biotechnology? Example of a fertility drug produced using biotechnology. While you're searching for that, I'll just read out what has been sent on the WhatsApp group. That the techniques used for immobilized plant cultures, they include the entrapping in an inert substratum, such as alginate, agarose, or polyacrylamide gels, absorbing onto an inert substratum, covalent bonding onto a biological macromolecule, embedding or entrapping in macroporous carriers such as polyurethane and such. Hence, based on that information, the technique that is not used for immobilized cell cultures is absorbing through biological macromolecules. That's then the answer to number 79, absorbing through biological macromolecules. All the other techniques, entrapping in an inert substratum or adsorbing onto an inert substratum, they are used, and also covalent bonding onto a biological macromolecule is used, embedding or entrapping in macroporous carriers is used as. Yeah. So that answers number 79. Thank you for that. Thank you for that, Dr. Sam. So on this, yeah, an example of a drug, of a fertility drug that is produced using biotechnology, given that we all went with choice A. So choice C. Follistim, yes, follistim, which is a synthetic version of FSH. FSH will stimulate the ovaries to produce eggs. Yeah, great. So follistim, it's a synthetic version of the hormone FSH, which will stimulate the ovaries to produce eggs. Just again, just to read out what has also been sent on the chat box, is that several fertility drugs have been produced using biotechnology, and one notable example is the recombinant FSH. We know FSH is a hormone that plays a crucial role in the development of follicles in the ovaries. In the past, FSH was extracted from the urine of postmenopausal women, but with biotechnology, we can now produce it using genetically engineered cells, which is more efficient and less prone to impurities. The recombinant FSH will be used to stimulate follicle growth in women undergoing fertility treatments like IVF. So additionally, other biotechnology-derived products such as HCG, GnRH analogs are also used in fertility treatments to help regulate the menstrual cycle and induce ovulation. Okay, so we have the recombinant FSH and others such as the HCG and GnRH analogs that are used especially for fertility treatment. Yeah. Moving to the next question, unless someone has something to add on that. I think throughout the discussions, at no point have we really dug deep, or at least done an overview of biotechnology. I don't know if at this juncture there's someone willing to just give an overview of biotechnology, from the vectors that are used, the techniques that are used, the purification, such things, the various steps of biotechnology. How do we obtain insulin? From the word go, how would you obtain insulin?
Okay, I can basically give okay, just a general, very brief, or kidogo tunachangia tu kidogo.
Yeah.
Content.
Okay.
Okay, so we usually have this recombinant DNA technique, and basically this procedure involves, from the word recombinant, we are recombining genes from two different organisms to make something we call transgene. So basically, organisms which we do this to, we usually call them or refer to them as transgenic animals, some transgenic organisms, sorry. Yeah, so usually, for example, we are trying to produce insulin. So what they do is they go to the human genome, they try and track which genes are responsible for the production of insulin. Then they obtain this gene. So how they obtain it is through there's a process of cutting using some genetic cutting materials. I don't know how we refer to them, but there are a lot of them. We have talents, we have I think the most common one that we use nowadays, it's CRISPR. It allows you to cut precisely at particular locations of the gene that you want. Then once you've obtained the genes that you want, you look for a vector. Basically, a vector will be the material or the transporter, what will transport to your genes and propagate them. So most common things that we use are usually the plasmids. So we usually take a plasmid as well, which is usually the extrachromosomal DNA of some found in some bacteria. So basically we take them as well. You splice them. I mean, you cut them first, certain regions which are usually close to the start codons and genes which are actually called open reading frames, where they're usually read. So you cut a section of that DNA using the same the enzymes you use initially, like the CRISPR or talents. There are usually other two, I think. Zinc fingers. Yeah, we have zinc fingers, etc. So once they've been cut, that region has been cut, they mix this DNA together, and then they usually add an enzyme. We usually call this enzyme ligase, which basically allows the transgenic gene, or this gene that you're adding to the vector, to be contained. They basically are able to stick to them. So once they've been joined together, we take this vector material, if it's the plasmid, then we put it in a place that will replicate. So usually, because they don't replicate by their own, we'll put them in certain bacteria, say E. coli, which will replicate. We give them enough nutrients and whatever enzymes that they need to replicate. So we leave them to replicate. They propagate our gene basically by multiplying. Then from that, during the transcription processes, they'll be able to produce the product that we put them to produce. That is just in recombinant genetic technology. So it's just an overview. Some other that we use, because I mentioned plasmid, we usually have some other vectors can be used, some viruses, particularly the adenoviruses. And I'm not sure if we use phages. I haven't. Okay, I'm not sure if we use phages, but I think we usually use adenoviruses as vectors as well. Sometimes we just directly inject that gene using micro needles. I don't know what they're called. There's those special, very tiny needles that are used to inject the gene that was already made into an organism. And then that product can be produced into the organism themselves. I think I've seen that with, I think, was it hemophilia? I don't know. Yeah, but it's sort of still upcoming technology. So yeah, just general overview.
Thank you so much, Dr. Thank you so much for that. You've really summarized the whole thing. Just to read out what has been sent on the chat box, although has already been mentioned, is that we have the restriction enzyme, restriction endonuclease. Okay, majorly they're just called the restriction enzyme. They serve to cleave DNA into fragments at or near specific recognition sites within molecules, known as the restriction site. So essentially, the restriction enzymes are the ones that will cut the required part of the DNA, and then ligase will join that DNA with the host DNA or with wherever you wanna fuse it with. Yeah. But thank you for that. Thank you for that. Does anyone have something to add on the same? Perhaps you've had a concept that you feel you can add a bit more. Just to check out the diagrams on the group. It's exactly what was being described. A synthesis of that insulin. We have the bioreactors from which we produce the insulin in bulk. Yeah. So I believe that is it. There's no one else has anything to add. We can move on to the next question, which asks the measure of the body's ability to eliminate a drug is called what? The measure of the body's ability to eliminate a drug is called what? Absorption, metabolism, clearance, or excretion. the measure of the body's activity to eliminate a drug. What is that term called? I'm saying clearance. Everyone is saying it's clearance. Or on pharmacokinetics generally, I believe we've discussed absorption, distribution, metabolism, excretion. So just a simple question on this is I think there's a formula for clearance, how you calculate clearance. What's the formula for that? If I'm not wrong, I think there's a formula for clearance. So what's the formula for clearance? If you're told to calculate the clearance of a certain drug, you're given its distribution, its whatnot, its various parameters. How would you calculate clearance?
So from the internet, it's written clearance, which is given in units of L per hour, is equals to the elimination rate over the concentration. So the elimination rate is in mg per hour, and the concentration is the plasma drug concentration in mg per L. Let me take us to the group.
Perfect. Thank you. So elimination rate over the concentration of the drug, you'll be able to calculate the clearance of a certain drug. Yeah. Just read out what has been sent on the group. So yeah, essentially the rate of elimination over the concentration. Yeah, the rate of elimination over the concentration. Exactly what Sarafina was saying. The rate of elimination over the concentration, you'll get the clearance. Yeah. Any other relevant pharmacokinetic formulas? How do we calculate the volume of distribution? We've discussed the things like first order metabolism, zero order, but how do we calculate volume of distribution? Someone is asking what's the difference between clearance and excretion? What's the difference between clearance and excretion? What's the difference between clearance and excretion?
So I'm saying excretion is the removal of drugs from the body, either as a metabolite or an unchanged drug. But when it came to the definition of the drug clearance, it's the rate at which the drug is removed from the plasma divided by the concentration. So maybe the difference could be excretions, like the drug is removed either as a metabolite or as an unchanged drug, and then there are different routes of excretion. But then clearance is how much of the drug has been removed from the plasma. Not sure if that's a viable difference.
I think that's right.
Okay.
Perfect. So that's the difference between clearance and excretion. Seeing another message. Kindly repeat. Saru, kindly repeat.
Oh, I'm saying for excretion, I'm saying it's been written that it's the removal of the drug from the body, either as a metabolite or an unchanged drug. Whereas clearance, it's indicated like it's the drug removed from the plasma, divided by the concentration of the drug that is in that plasma. So maybe the clearance is specifically removing the drug from plasma at a certain rate, while excretion is removing the drugs from the body, and it can be removed either as a metabolite or an unchanged drug.
Okay. Hiral, we good?
Yes, thank you.
Perfect, perfect. So distribution, no one has said how you calculate volume of distribution.
It's been sent to the group. Volume of distribution is equal to the amount
Oh.
of drug in the body divided by the plasma drug concentration.
Nice, nice. Amount of drug in the body over the plasma drug concentration. Great, great. Then even now, you can be able to calculate the elimination rate constant when you've calculated clearance and you've calculated volume of distribution. So all of these are interrelated, right? So I'm seeing on the chat box. I believe, yeah, this was sent, yeah, as an image to the WhatsApp group. We'd already answered that. Moving on to the next question, unless someone has something to add on the same, on pharmacokinetics in general. Review the answer. Could you, Savin, could you go back to number 79? Mobilizing can be achieved through entrapping, absorbing through covalent bonding involved, which may not be suitable for immobilizing or other cells in some cases. Yeah, so there are two different references which are alluding to two different answers. One was alluding to absorbing through biological macromolecules. Now this one is alluding to covalent binding onto a biological macromolecule.
I should be see from what Sam sent in the group. Literally it's written there. There are different ways you can immobilize. The techniques used for immobilizing plant cell cultures, and A, B, and C are all there. And when it comes to the absorbing, it's absorbing onto an inert substratum. There's nothing written about absorbing through biological macromolecules. For macromolecules, it's written covalent bonding.
Now, according to the answer that, or rather explanation that AI has sent, it's saying that there is such a method such as absorption through biological macromolecules.
Okay, yeah.
Anyway, on this, as previously what we've done when we're having two sources saying two different things, we just put a star, we come back to it when we come back to it. We probably won't, but we just put a star so that everyone is relaxed. We can continue. To number 82. So number 82 asks which of the following statement best describes a drug's half-life. Which statement best describes a drug's half-life? This one we've done this a million times. amount of drug required to achieve half therapeutic effect, time required to change the amount of drug in body by 1/2 elimination, time required to absorb the drug by 1/2 at the site of action, or amount of drug required to absorb the drug by 1/2 at the site of action. For that is choice B. Right, time required for the amount of drug in the body to reduce by half or to fall by half. We don't have to dwell so much in that question. We've described the half-life before. So moving to the next question, the question says the fraction of unchanged drug reaching systemic circulation following administration via any route is termed as what? Fraction of unchanged drug reaching systemic circulation. Is it bioavailability? Is it bioequivalence? Is it volume of distribution? Or is it the therapeutic index? Fraction of unchanged drug reaching systemic circulation.
Choice A.
Bioavailability, perfect, yeah. So what is bioequivalence? We've talked about volume of distribution before. And what is therapeutic index? Yeah. So what's bioequivalence and what is therapeutic index? Anyone can unmute and
For by equivalence, it's usually basically a relationship between, usually related to one drug to another, basically by comparing the mechanism of action, the bioavailability, the clearance, basically the pharmacokinetic properties. If administered to the same drug, say you have drug A and then drug B. So basically, if drug B has the same, if administered through the same route, has the same mechanism of action, the same okay, basically it's just the availability, the same clearance, the same availability and clearance basically is what we'll be looking at mostly. If they are equal then, or almost equal, then we say that by equivalence. Usually, I think it's usually done for drugs, say a branded drug, an original, then for the generic drug. So usually they usually look at a range of between 85% to 115. If they're actually within that range, then they're termed as bioequivalence. Correct.
Thank you, Sam. Thank you, Sam. Yeah, so yeah, bioequivalence as described is that property where the two drugs, I'll read from what Stanley has sent on the group, that bioequivalence is the property wherein two drugs with identical active ingredients or two different dosage forms of the same drug possess similar bioavailability and produce the same effect at the site of the physiological activity. So essentially bioequivalence, yeah, this is where we are having two brands of the same drug, for example, Panadol and your generic, your normal paracetamol. Let me not say generic, all drugs are generics. So Panadol and your another brand versus another brand of paracetamol 500 mg, we'd say they are bioequivalent. They are having the same active ingredient and they possess the same bioavailability and will produce the same effect at the site of activity. So just to read out the other stuff related to bioequivalent studies, we've looked at bioavailability, just the fraction of drug that reaches systemic, the fraction of unchanged drug that reaches systemic circulation. Looking at pharmaceutical equivalence, these are drug products containing the same amount of active ingredients, but not necessarily containing the same excipients. So pharmaceutical equivalence, drug product containing the same amount of active ingredients. Same amount of active ingredients. If it's your paracetamol, they both contain paracetamol, 500 mg, but not necessarily containing the same excipients. Good example, perhaps, in my limited knowledge would be Cipladone 500 mg and a paracetamol 500 mg. They are containing the same amount of active ingredients, but we know the excipients are not the same. One is made to disintegrate, the other one is made to be effervescent, the other one is not. Pharmaceutical alternatives, these are drug products containing the same active ingredients but different salts, right? Same active ingredient but different salt. For example, tetracycline hydrochloride and tetracycline phosphate. It's the same active ingredients but different salt. Remember, for the equivalent was same active ingredient but different excipients. Alternatives, different salts. Generic equivalents, on the other hand, these are drug products containing the same active ingredients but different brand names, provided that the dosage form is the same. That is Amoxil and example of Wimox, right? We have the same active ingredients but different brand names. So just to correct what is happening in the community practice is that there is no such drug as original. That is, that term is not correct. The right term is originator brand or originator. Yeah, that was the first brand. It's the originator, but it's not original. Because all drugs, we know all drugs are generics. We learn drugs in their generic names, not in their brand names. Just pharmaceutical equivalence is not equal to bioequivalence. Pharmaceutical equivalent is not equal to bioequivalent. Okay. Then for the next photo, just some factors that would affect bioavailability. We have the physiological factors, the physicochemical properties of the drug. Under the physiological factors, we have the pH, right? pH here, majorly we're talking about factors affecting bioavailability under the drugs that are administered through the oral route. We have the pH, of course, the surface area of the intestines, the presence of carrier proteins for absorption and exsorption. We know the P glycoprotein actively pumps out drugs and other substances out of the cell into the GI lumen. Enzymes here, endogenous and bacterial. This is the reason why we cannot give insulin orally, right? To be digested by these enzymes. GI blood flow, of course, plays a role. The GI blood flow has to be adequate so that the drug can be able to reach the systemic circulation. Gastric emptying and intestinal transit. Food would delay gastric emptying. Then looking at the physical chemical properties of the drug, the pKa, the water and lipid solubility, the molecular size, stability in the GI environment, specificity for carrier proteins and enzymes. So we look at the physiological factors, physical chemical properties of the drug. We can even have patient factors such as presence of a disease. For example, a disease, if a patient is having diarrhea, that would definitely affect the bioavailability of your drug. On the other image, just more definition of the terms. If ED 50, that is effective dose 50, this is the dosage of the drug that is effective in 50% of the test population. So for example, if it's an antihypertensive, it's at a specific dosage, we find it will lower the blood pressure for 50% of the population. Lethal dose 50, same thing, only that fatal toxic dose, same thing, only that now we are manifesting the adverse drug reaction, the toxic effects. Then the therapeutic index, we know it's that measure between, we divide LD50 divided by ED50 to get the therapeutic index. Lethal dose 50 over effective dose 50, we get the therapeutic index. Just something to note is that you can see the lethal dose is the numerator and the effective dose is the denominator. That's why we say the therapeutic index, the bigger it is, the better, because the bigger it is, it means there is a big difference between the lethal dose and the effective dose. So imagine if the lethal dose is 2 milligrams and the effective dose is 1 milligram, right? That would make this drug have a therapeutic index of 2. But imagine for the same drug, let's say 20 and 10, yeah? Let's say 20 and 10, it would still make it 2. But imagine for the same drug whose lethal dose is 100 mg and the effective dose is still 10 mg, it would be 100 divided by 10 and would get the therapeutic index of 10. Yeah, so we want the therapeutic index to be as large as possible, right? The therapeutic window, we know this is that concentration between the minimum effective concentration or the minimum therapeutic concentration and the maximum tolerated dose or the minimum toxic concentration of the drug. Is that window, that range of concentration between the minimum effective dose and the minimum toxic concentration of the drug. So as mentioned yesterday, that narrow therapeutic index drugs require routine plasma or serum drug monitoring. This is where clinical pharmacists come in. Narrow therapeutic index drugs, they will require that therapeutic drug monitoring. Yeah. And while we are at it, you can be mentioning on the chat box the various examples of narrow therapeutic index drugs. Yesterday we were told one, which was digoxin. On the next image is even there. You guys are lucky. You guys are lucky. So yeah, it's just that definition. Narrow therapeutic is a very close margin between the concentration in the blood of circulation and yeah, so it's sorry for that. That concentration between the narrow, it's the close margin between the concentration of blood in circulation of a drug that is therapeutic and that concentration that is lethal. So there's just a narrow therapeutic index. That safety margin is so small, you can't afford to mess up with such a drug. Yeah, it's not like your paracetamol that has kind of a wide therapeutic index that if someone has overdosed, you still tell them, you'll be fine. So moving on to the next question.
I have a question.
Yeah, go ahead.
Is therapeutic index and window the same thing, or there are different things?
So is there Go ahead.
I'm saying I think they're almost similar. Like
according to my understanding that it is the same thing, but you have defined them as different things. Okay, different. I have given them different explanation.
Okay, it's the same thing.
But let me read what I'm getting. So the difference between therapeutic window and index. So the therapeutic index can also be looked at a slightly different way, and then it will be called the therapeutic window. What the window means is as the dose increased, first you get to the effective dose, and if you keep increasing the dose, you eventually get a toxic dose. So I think so we've said the therapeutic index is the difference between it being effective and toxic dose. So the first thing you start with is the effective dose, then you when you keep increasing the dose, you eventually get to the toxic dose. So it's almost the same thing, I guess. But the window I guess it shows the progression where you move from the effective dose all the way to the dose where it's toxic.
Okay, the two terms are the same thing, actually means the range between above which the toxic effect is felt and below which there will be no therapeutic effect. So it's actually the range for both the explanation.
Okay. If it
Okay.
says so.
Yeah. So yeah, only that perhaps for the index is more of that lethal dose over effective dose, and minimum effective to minimum toxic dose. You know, it's just these are kind of the same thing. Yeah, as Pablo has said, yeah, index is a ratio between that and that. Therapeutic window is a range. Otherwise, they describe the same thing. Yeah, that difference between when a drug is safe and when a drug is toxic, just that difference. Moving on, unless someone has a question, addition, something to add on the same.
Okay, I have something to add that I read somewhere some time back that most drugs are monitored via the trough, something to do with the trough. Yeah, and a few drugs are monitored using both the peak and the trough. I don't remember the specific drugs.
So saying that some drugs, we know how that concentration will fluctuate, peaks and troughs, that some drugs will be measured using the troughs, other ones will be measured using the peak and the trough. Yeah. So if someone can shed more light on that, yeah. So something has been shared on the group about the same, or rather about what we were saying, the therapeutic window, just without those minimum, maximum something. Is just the therapeutic window is that range between the desired response and when the adverse responses start kicking. Yeah. Duration of action, peak of effect. So yeah, can just proceed if perhaps there's no addition. Just to read out one last thing, that in simple, therapeutic index is the difference between the maximum toxic dose and the minimum effective dose. Maximum toxic dose and the minimum effective dose. So yeah, moving on to number 84. Which one of the following factors least affects the rate of drug metabolism and elimination? Which of the following factors least affect the rate of drug metabolism and elimination? Age, sex, time of administration, or liver size? Time of administration. Are we all good, Hapo?
Yeah.
So number 85, which of the following routes of drug administration is most effective in avoiding the first pass effect among the ones listed? Which one is most effective in avoiding the first pass effect? Is it the sublingual, rectal, oral, or inhalation? Which one is the most effective in avoiding or bypassing the first pass effect?
Wouldn't it be rectal?
Rectal. rectal compared to sublingual and inhalational. Rectal, 50% of rectal, they reach the portal circulation, hence undergoing fast metabolism.
Okay.
Yeah. So between sublingual or inhalation, I think we're having sublingual, someone is saying sublingual. Because in
Excuse me,
sublingual, they
I was going through past papers, so there was this one that had been marked sublingual, right? And I googled, so sublingual was right.
So sublingual is the correct one.
Hello.
Yeah,
To
hello.
provide an explanation to that, it's because you see the blood that is in the vessels that supplies the sublingual area does not pass through the hepatic portal circulation, and that's why.
Perfect.
Yeah.
Yeah, so the blood that supplied that sublingual area does not pass through portal circulation, it just goes directly to the bloodstream. In inhalational, I know there's an aspect of a very small aspect of fast pass metabolism through inhalational route of administration. There are some drugs that can be metabolized in the lungs. So someone is requesting that we go through other routes that avoid fast pass metabolism. So we can just shout them out. Other routes that avoid fast pass metabolism, same IV, it's been listed as number 1.
Yes. So
We
if
should
they
just
had,
say parenteral
if
as
they
a
had,
whole. Parenteral as a whole.
Intramuscular.
Intramuscular, so Lydia said just parenteral as a whole. That's
okay. So does
that as a whole.
does
The
transdermal
first part.
count under
Yeah.
parenteral?
Yeah.
Okay. And I'm seeing
as long as it's not where the GI.
I'm seeing also suppository.
But let me ask, for this question number 85, what if they had IV as one of the choices?
I think IV is the
most
standing. Yeah. Yeah. So yeah, parenteral as a whole, and the various stuff. I think there was an image that had even been sent regarding those parenteral routes. Someone is asking, does rectal go through fast pass metabolism? Does the rectal route undergo fast pass metabolism? So I'm currently looking for where I read that, but
I think it does, because there's these guys who do butt chugging, they're taking alcohol, and it's not advice like a guy.
Yeah, because I think there's an inferior rectal vein, that one will take it to systemic circulation, but there's another vein that will also supply in the rectal area that will take it to the liver. So yeah, but only a third. Yeah, only a third. Yeah, so the rectal does go through fast pass metabolism, but only a fraction of it. Thank you. Moving on to number 86.
Excuse, I have something
Drugs
to add.
are.
Excuse. Someone just asked if transdermal is a parenteral route, and that there's a discussion where we say that transdermal is not a parenteral route of administration. Just wanted to clarify that.
But there's actually a difference between transdermal and intradermal. I think that is.
I think she asked about the transdermal. if it's a parenteral route of administration. And there's a discussion where we say transdermal is not a parenteral route of administration because you don't use a needle to administer the drug.
Yeah, correct.
Perfect. Thank you for that. Yeah. So question number 86, I'm seeing just Thomas is adding that transdermal patches are used. Transdermal patches are used, just as an addition to the same. So number 86. Just to read out something that has been sent.
Sam is asking a question. He said, what was the thing about taking alcohol again?
I think that one they'll discuss with Chai.
It's okay.
On their free time. Yeah, but just to read out what has been sent regarding rectal administration. In general, drug absorption in the upper part of the rectum is transported to the liver via the portal system, and thus undergoes fast-pass metabolism, whereas drug absorption in the lower rectum is transported directly to the systemic circulation. So just as we've seen here, there's a fraction of the drug that will undergo and a fraction of it that will not undergo the fast-pass metabolism. We're seeking clarification on whether transdermal avoids fast-pass metabolism. I think transdermal does avoid, yeah. Yeah. So to the next question. Drugs are transported across biological membranes through all the following means except drugs are transported through membranes through all following: active transport or osmosis. So drugs are transported through all of the following mechanism across the biological membranes except active transport, osmosis, passive diffusion. Yeah, filtration. Yeah, so filtration is that we see filtration mostly in the blood or the kidneys, yeah, and all such areas. So filtration it is. Then a question is, what's now the most common way through which drugs are transported, especially from the GIT into the enteral cells? What's the most common way through which drugs will cross that biological membrane? Is it passive diffusion, active transport, or osmosis?
Is it active transport?
Transport. Someone is saying active transport. Anyone else? I think it's passive diffusion.
Passive diffusion.
Yeah, passive diffusion. Yeah. Great. Moving on to number 87, unless someone has something to add to the same transport mechanism of drugs. Yeah. Or sorry, what? Something perhaps that we may need to discuss just briefly. This is more physiology than pharmacology. Is types of transporters, the antipodes, the sympads, how just those types of transporters. Someone is asking, passive diffusion requires a transporter. Not it doesn't. Facilitated diffusion, I think, is what requires the transporter. Passive is just passive. Yeah. So facilitator is the one that will require a transporter, though the mechanism is still diffusion. So examples, or not example, types of transporters in physiology or in pharmacology, the types of transporters. Who can tell us the types of transporters? The types of transporters in the body, types of transporters. In our whole discussion, we've been mentioning a lot about the sodium potassium ATPase. What type of a transporter is it? So it's active plasma that requires transporters, ATP transporter, uniport, simport. Dr. Hasna, perhaps you can explain further. What's a uniport? What's a simport? What's an antiport? Yeah. And then or anyone who has seen those terms and remembered those transporters. What's a uniport? What's a
I can explain one. I can explain the simport one. It transports two molecules in the same direction. An example is the sodium glucose sodium glutathione transporter in the intestinal lumen, where it transports sodium along with glucose in the same direction. Yes.
Thank you. So simport, two molecules, same direction. Nice. For example, the sodium glucose transporter in the intestines, even in the kidneys, to transport sodium and glucose into the cell. So simport has been described. The other one, you can just pick one, describe it, give us an example of it.
And then uniport, it transports one molecule. Example is GLUT4 in the kidney. I'm not sure the location, but the kidney, which one? The one that is involved in it transports glucose from the, I'm not sure in the direction, but it transports the GLUT4, the one which the glifloxacin acts, the drugs. Yes, that one is a uniport.
So a uniport, yeah, this transports one molecule in that direction. For example, it's talked about the GLUT4, right? Just transports glucose in. No, it doesn't need any other need. Just to read out, someone was saying the differences between uniport, simport, and antiport. The uniport, it's an integral membrane protein which transports a single type of substrate species across the cell membrane. The simport, also integral, will transport two different molecules in the same direction. The antiport, also an integral membrane, which will be involved in secondary active transport. Emphasis on the active. We are seeing the antiport, there's that
Antiports involves movement of molecules in the opposite direction. Example, expulsion of hydrogen ions using the sodium ion gradient in the kidneys.
So I think Gaius was mentioning that the antiport has to be active because you're moving it across a gradient. So I think when you're moving anything across a gradient, there has to be energy that is needed. That's why it's antiport is used in active transport. Yeah.
Even symport is active. Apart from whatever, all others are under active transport. Under active transport, we have primary active transport. Under that itself, we have ion transporters and ABC ATP binding cassette transporters. Then we have secondary active transport. Under that, we have symport, that is called transport. We have antiport. So I don't think if uniport is under active. Have you gotten? Watu wako wapi?
Tumepata, sorry.
Okay. Great. So those differences have been shared.
Gaius,
Yeah.
ni mbona sio osmosis? Juu filtration, an example could be kama alcohol. Hebu nipe example ya osmosis. Juu mumesema filtration dancer. Mimi nasema osmosis.
But it's asking about drugs.
Yeah, alcohol is
which example of drug is transported by osmosis? Ndo nauliza. I thought osmosis ni movement of the solvent, not the solute. And I think drug ingekuwa solute. Okay, we could think out loud.
Yeah, and even osmosis is only the movement of water.
So filtration or osmosis. But is osmosis the movement of water or of solvent molecules? Is it specific to water?
It's not specific to water.
Solvent molecules and some drugs that are in solution form, so not necessarily water.
Solvent.
Trevor has a song Maji ni Dawa.
So we need a clear answer on that. So mechanics of drug absorption. Filtration or osmosis? Perhaps Black Panther can tell us about filtration. What's an example of a drug that is absorbed through filtration?
Alcohol,
Oh, filtration. How does that take place?
See, when it moves from, okay, meaning in terms of time ya ku excrete. Yeah, see, it's still transport.
But as we've looked at it, majorly like in absorption. Drugs are across.
Si wame specify to transport, hawaja specify.
True. Across biological membranes. Yeah, they haven't specified.
Blood pressure, like. Also from blood to tissues, alcohol will move through filtration.
Raleigh is saying that she doesn't.
So, because I'm saying alcohol is absorbed from the stomach and small intestine by diffusion.
Okay, from the blood.
No.
So we can go with filtration as of today.
Hata kuna mahali wanasema answer ni osmosis kidogo tu, acha nisha.
Having someone saying when you filter, whatever you are focused on doesn't pass through.
So the answer is osmosis.
It would appear so.
Okay.
Isn't osmosis part of transport? Because when we talk about filtration, you just like when we talk about filtration, I think it would be like in the kidney, but that's not transport.
What do you define as osmosis? I think it being through a membrane is part, a semi-permeable membrane is part of the definition. Passive
Exactly.
diffusion can happen even in a room. Place a perfume in a corner, it will diffuse to other parts of the room.
Oh, then passive diffusion makes sense. Because if we're talking about across biological membranes, then filtration would be across biological membrane. If we're talking about it in, for example, in kidney.
But we've said most drugs are absorbed through passive diffusion, and that is movement through the biological membrane. Some were waiting for your reference.
Osmosis makes sense.
Correct.
Osmosis makes sense actually. So, because osmosis is transport of solvent molecules, not solute, and drugs are solutes. So yeah, osmosis.
Isn't osmosis a form of passive diffusion? I'm just asking.
No. Oh, maybe yes, it is. But of solvent molecules. So, and drugs are not solvents, drugs are solutes.
Somebody said water is also a drug.
This is Jibas.
And a solution is different from a solvent. So drug dissolve me solution. So the answer is osmosis.
So nimetuma the reference. Okay, ni from Bing, but yeah, basically na rule out the other answers. You can just have a look.
Great. Let me just, so filtration is the movement of drugs and water through pores in the membrane driven by the hydrostatic pressure across the membrane. It does not depend on lipid solubility or ionization, but rather on the size of the drug molecule and the pore. Drugs are small and water soluble, that can pass through the active transport. I don't think you need to dwell much on that, because active transport, we already accepted it's an answer. Passive diffusion also, we don't need to dwell much on the same. Osmosis is the movement of water across. Osmosis does not involve the transport of drugs across biological membranes, but rather the movement of water to balance the osmotic pressure. Okay, so we can go with osmosis. Yeah, because
Guys,
of
yeah, if we can agree that there is no drug that is a solvent, then we can go with osmosis. But if there is, of which I'm thinking there is, then should be filtration, because when you're filtering, you're eliminating nothing, as in let's say a certain solute X. If you're filtering it, then it doesn't go through. So there's no way you can say a solute X or a drug X transported through filtration. You have filtered it, not gone through.
Yeah. Yeah, by the way, Sam, respond to that. Filtration. When we say filtering, it's through a pore. It's essentially through a hole, not across a membrane. That's a very nice, and even your explanation on being says the same. It's through a pore, not through a biological membrane.
The pores are in the biological membrane, or
the pores are not between cells, between cell A and cell B.
which are in the
Pores are
membrane.
between cells. No, they're between cells, not membranes.
So they're not in the membrane?
No, they're between two cells.
So I think you're complicating it. As long as it is from site A to site B of the membrane, as long as it is across, that is across the membrane.
Now, there's something that has been said saying that lipid insoluble drugs cross biological membranes by filtration if their molecular size is smaller. So I think on entire question, we just, as the presenter has done, just leave a star to be discussed further. And Pablo is saying the purpose of filtration is separation of solute and solvents, not transportation. Anyway, we've spent enough time on that question. So moving on to the next. In drug biotransformation, which of this reaction involves the conjugation of the drug or its phase 1 metabolite with an endogenous substrate? So in drug biotransformation, which reactions involve conjugation of the drug or its phase 1 metabolite with an endogenous substrate? So in short, which one is a phase 2 type of reaction? Can I agree that it's acetylation, right?
Yes, because you add an acetyl group to the molecule.
Yeah. So acetylation there. Phase 1 and phase 2 reaction, we've looked at them before. Phase 1, the ones involving the CYPs, right? Oxidation, reduction, even hydrolysis, we classify it as a phase 1 reaction. Then phase 2, that conjugation, glucuronidation, acetylation, sulfation, all those phase 2 reactions. Unless someone has something to add on that or a question.
Yeah,
I have something in the previous question.
I don't know who. Okay,
Lydia,
go ahead.
go on.
Yes, can I read?
Yeah, please do.
Okay, let me just read instead of this. Okay, let me find under osmosis. Water-soluble drugs can move across membranes through water channels into brackets aquaporin. So osmosis is one of the process of drug transport. So the other is filtration.
Okay. water-soluble. She said that water-soluble drugs can utilize the aquaporins to move from side A of the membrane to side B of the membrane across biological membranes. So the answer is filtration based on that explanation.
You said that water soluble drug is water. Like the fact that it is water soluble, it's actually a solid that is actually dissolving in water. So osmosis still remains the movement of water. Like, why are we settling at that? Because that is a water soluble drug. It's not water.
Can I repeat the explanation?
Yeah, please.
Let me just give me a second. So the osmosis, so the water-soluble drugs can move across membrane through the water channel. So they use the water channels by osmosis to move across the membrane. Okay, that's all.
Okay, did they say that they move by osmosis in the water channels? Or they just use the water channels?
That's from Google, not my
Okay.
That's my opinion. That's from Google.
But I thought, okay. Substances move across the aquaporins. What is the process? I thought it was endocytosis.
I think, guys, for that question, let's just leave it at that, because also something else shared on the group we'll see simple diffusion, filtration, osmosis. So that one has both filtration and osmosis. So we can think we can leave it at that so that we move on to other questions. Yeah.
Okay, and on question 87.
Mm-hmm.
Yeah, I was asking about phase 3 metabolism. Yeah, because there was something like that mentioned. Maybe people in my class can shed some light on this. I remember it was through the the ABC, ATP binding cassette and something else, and I'm not sure how. Yeah, but then there's something like phase 3 metabolism. Yeah, Lydia?
I have no idea.
And anyone else with an idea about phase 3 metabolism?
It's okay. Maybe we can just move to the next question.
Okay. Good. Okay. Let me just read. Dr. Irala sent something that drugs can ultimately undergo further metabolism during phase 3. In this phase, the drug that gets transported via an ABC transporter, which will require ATP, which will require energy consumption to actively uptake or efflux a compound from one side of the cell membrane to another. So she's shared something about the ATP binding cassette superfamily of transport proteins, whereby they will hydrolyze ATP and function as an active efflux or uptake transporters. Okay. Then on aquaporins, again, as we said, we're going to skip that so that we don't spend a lot of time for such a question. Moving on to number 88. Dash occurs when the rate of drug elimination remains constant irrespective of its concentration. This one we've discussed before, right? Drug elimination remains constant irrespective of the concentration. That would be zero order kinetics. Doesn't depend on the concentration is just a constant amount of drug that is being eliminated per unit time. Unless for first order, we said it before, does anyone know what exponential kinetics is and what constant rate kinetics is? Perhaps just to define the terms that are on the choices there. Exponential kinetics and constant rate kinetics. What do those terms mean? Or do those terms exist? And if they do, what do they mean? Exponential kinetics and constant rate kinetics. Just an addition to the previous question, that phase 1 will occur in the endoplasmic reticulum, whereas phase 2 will occur in the endoplasmic reticulum of hepatocytes, whereas phase 2 will occur in the cytoplasm of hepatocytes. Dr. Hasna is telling us that exponential is also known as first order. So first order, aka exponential. What about constant rate? Anyone, what's Google saying when you search constant rate kinetics?
So constant rate kinetics refers to the type of chemical or enzymatic reaction in which the rate of the reaction is constant and independent of the concentration of the reactants, while the exponential rate kinetics is whereby the rate is directly proportional to the concentration of the drug.
Constant rate. It's sounding like zero order, but anyway, no comments.
Hello, from understanding only from only what he has read. Order, maybe if you're eliminating many 50 the 50 of the drug, even if you increase the concentration, will keep on eliminating 50. But exponential, if you increase the concentration, the elimination will also increase. It will keep on changing depending on the concentration. That is first order or exponential.
Okay. Okay. Nugali saying constant rate is zero order. Constant rate is zero order. So moving on to the next question before on therapeutic drug monitoring. That it will be it will be useful in all the following situations except drugs with a low safety margin, potentially toxic drugs used in the presence of renal failure, in cases where drugs reach high plasma concentration within a short duration of time, in cases of poisoning. So TDM, therapeutic drug monitoring, we use it for in all of the following circumstances except which one? TDM. TDM, therapeutic drug monitoring. Therapeutic drug monitoring.
Let me ask, if a drug reaches high plasma concentration within a short duration, does it mean it's easy for it to become toxic to the person?
It will depend on its
No.
therapeutic index.
Yeah.
Okay. So will that be the answer?
I think if the drug reaches high plasma concentration within a short duration, it also means that this drug, it can be quickly eliminated from the body. Because rapid rise and fall in the drug concentration may lead to like quick onset of therapeutic effects, which also leads to it won't lead a prolonged time. It won't be for a long time. It will be eliminated quickly. I think that's the answer.
Yeah. So choice C, in cases where drugs reach high plasma concentration within a short duration. Are we all in agreement with that? If you find any information that perhaps is different or any additional information, you can share and we will discuss it here. Moving on to the next question. On pharmacotherapy, the requirement of a higher dose to produce a given response is referred to as tolerance, resistance, efficacy, or potency. Higher dose, same response. Tolerance, resistance, efficacy, potency. Hilal is saying that is tolerance. Yeah. So there we are on agreement. We've looked at tolerance, tachyphylaxis, potency, efficacy. We've discussed all those details before. So we can move on. Unless someone has something to add on the same or a question on the same. Perfect. Descriptive statistics. We mentioned this yesterday. Patient is asking what's potency? What is potency? What is potency? When you say this drug is potent, what do we mean? What do we mean?
So, according to Google, potency is an expression of the activity of the drug in terms of the concentration or amount of the drug required to produce a defined effect, which means, for example, a drug which gives a high effect, a drug which little amount of the drug gives high effect means it's a high potency drugs. When you need to use a lot of the drug in order to achieve the desired effect, it means the drug has a low potency.
So to add what she has said, potency is the strength or concentration of a drug required to produce a specific biological effect, and it measures how much of the substance is needed, while efficacy refers to the maximum effect or response that a drug or compound can produce and does not depend on the concentration or the dose. It is just the inherent ability of the substance to elicit a biological response. That is efficacy and potency.
Great. So we've differentiated efficacy and potency, assuming we have, for example, this is an example. Don't take this out of context. We have losartan and telmisartan, for example. Oh, let me just say drug A and drug B. Two antihypertensive drugs, drug A and drug B. So for them to lower pressure by 25%, drug A, we need 2 mg, drug B, we need 50 mg. So that means drug A is more potent, right? Lower concentration, same effect, right? Drug B, low potency, We need 50 mg to elicit the same effect. But efficacy is the ability to, at the end of the day, reduce, for example, now in our case, we've said reduce the blood pressure by 25%. Both drugs are efficacious, right? They will both reduce the pressure by 25%, but one is more potent. We just need a little bit and it will do the job, as compared to the other one. We need a lot of it to do the job. So efficacy is ability to do the job. We don't care about the amount of drug. Potency, now we are like, hey, which one do we need in a lot of amounts that is a low potency? I hope I've Srina?
So potency is correlated with affinity. From what you have explained, potency is correlated with affinity. The higher the more potent the drug is, the higher its affinity.
Hamimti, go ahead.
Yeah, I wanted to say efficacy, as you already explained. When you say a drug is efficacious, that means the degree at which it can work. For example, if you compare two analgesic drugs, opioids are analgesics, and also paracetamol is an analgesic. But opioid, it is an analgesic, at the same time it is euphoric, at the same time it is respiratory depressant. Like that is efficacious. Like that one, it is an efficacy. For potency, we just dwell on the concentration of the drug. For example, the digoxin is very potent because like micrograms, like you can use micrograms to elicit the pharmacological action. Yeah, that's what I can say.
Perfect. So yeah, then on the group also saying the group also, I'm seeing something has been shared on the same. Efficacy is the maximum effect that a drug can produce, irregardless of dose, right? Maximum effect that a drug can produce. However, potency is the amount of drug that is needed to produce a given effect. So efficacy, effects that the drug can produce, irregardless of the dose. Potency, amount of drug needed to produce that effect. Then someone has sent something beautiful on therapeutic drug monitoring, saying that in theory, I'll skip to drugs that do not need, these are drugs that do not need therapeutic drug monitoring. We know not all drugs, actually most drugs don't need therapeutic drug monitoring. And examples of these are the drugs used for treating diseases of which their clinical endpoints can easily be monitored, such as blood pressure, heart rate, cardiac rhythm, such things. Drugs whose serum concentration do not correlate with their therapeutic effects, so that one you don't need to do TDM. Drugs with less complicated pharmacokinetics, and drugs used to treat less complicated or not life-threatening diseases. Those are the commonly monitored drugs, things such as theophylline, aminoglycosides. We know their toxicity is quite major, vancomycin, immunosuppression such as cyclosporine and methotrexate. Those are examples of drugs which require those therapeutic drug monitoring. Just to add on the same, that the other indications would be the drugs for which the relationship between the dose and the plasma concentration is unpredictable. A good example being phenytoin, drug with a narrow therapeutic window. That one we already said. Drugs with a steep dose response curve, such as theophylline. Drugs for which there is difficulty in measuring or interpreting the clinical evidence of therapeutic effects. Drugs with a saturable metabolism, again, phenytoin comes in, and drugs with a poorly defined endpoint or difficult to clinically predict the response, such as the immunosuppressant drugs, the likes of methotrexate, cyclosporine, as listed in the previous example. Yeah, the chat box, nothing on the chat box. Shrina, I hope you're good now. You can differentiate between potency and efficacy.
Yes, thank you.
Great. Descriptive statistics, right? Descriptive statistics. We spent hours on this in a previous day. So what is the descriptive statistics? Is it based on the principles of probability? Represents conventions for planning research, represent conventions for summarizing and organizing data, specify the selection of appropriate measurement skills. I think something was shared between descriptive and inferential statistics. We can all agree. Descriptive represents conventions for summarizing and organizing data, such as in graphs. Yeah. Moving on swiftly to number 91, unless someone would have a question on the same or an addition. Number 91, the question is, which one of the following is unique to the experimental research strategy? Which one is unique to the experimental research strategy? Assignment, selection of cases to be studied, definition of population, participant observation. So which one is unique to the experimental research strategy? Which one is unique to the experimental research strategy? Assignment, selection of cases to be studied, definition of population, or participant observation. My researchers, we have published authors such as
I think it's assignments. In experimental research, you have
Dr. Sonia
to...
here, perhaps they...
Hey guys, unanisikia?
Loud and clear, Dr.
Oh, so in assignment, it's where you have to, assignment is what is unique to experimental research. That's where you have to, like you have your different groups, then you have now to assign control ni gani, which you're going to give the treatment product, even those ones. Test ni gani, control ni gani. That's the assignment.
Okay, so assignment. Can move to the next unless there's someone of a contrary opinion.
Just as Seraphine said, experimental research involves assigning subjects or participants to different groups. And these different groups include control group, the experimental group, and so these other answers are usually associated with research studies such as case studies or surveys or observational research. So assignment is unique to experimental research such that you're assigning subjects to different groups.
Perfect.
I have a question. In the experiment, I thought experimental is where you go to the lab. Are there like participants or just the researcher?
Seraphine, back to you.
What's the question?
Experimental research strategy is where you go to the lab to do your experiment.
Yes.
So are there participants involved, or it's just the researcher who goes to do the whatever work he or she wants to do?
Oh, it depends, but even if it's in the lab, you have to assign, let's say you're working with mice, you have to assign them to the different groups. Or even in clinical trials, which are also a type of research, then you'd still need to assign.
So what I was trying to say that in experimental research design, there are no participants. We have the mice, but mice are not, okay, they're participating, but they're not participants. I thought participants are people.
Oh, she meant the experimental, it's not just on the lab. So she said the lab is where we use the rats and everything, yeah. But the experimental is not just in the lab, in that the one we do the clinical trials is also an experiment, in that we are trying to experiment the effect of a drug on human beings now. Is that okay?
No.
It's okay.
Number 91. The advantage of including a control group in a study of treatment effectiveness is that effects of maturation can be eliminated, reduced, measured, ignored. What's the advantage of including a control or a companion group in a study of treatment effectiveness? You are studying as if your drug has hypoglycemic activity and you include a control group. Why?
See.
Pardon?
See, the control group, because if the control group will be the ones that are not getting the drug, yeah? Are the ones getting the drug?
No, I think control group, you'll give them something that is already known. For example, insulin, that's a positive control. I think, hey, these things, what wala okay.
So yeah, then you can definitely measure, because if there's a control group and then there's a group which is not receiving and then there's a group which is getting the actual drug, you can easily measure the effect, I guess, using the parameters you had set.
So someone is asking, what is maturation? Just as a side note, what is maturation? The chat box.
In terms of what?
In terms of this question, in terms of the question here.
I think it's effects of maturation is at the end of whatever you're trying to do.
So in the case, just as an example, in the case of we are testing a plant with hypoglycemic activity. So just say how would maturation be there to bring the message home?
You see, like in this question, the advantage of including a control or a companion group in a study of treatment effectiveness is that effects of maturation, like at the end of your research or at the end of your study, you can be able to actually see the difference between the two.
Great. I think Sam has understood. Moving on to
me, I haven't understood, because what I'm getting in terms of maturations are other factors that you're not testing. They're contributing, but it's like things that you can that will happen in the course of the study.
I don't agree.
That's what I'm saying.
I don't get it like that.
Okay, how are you getting it then?
All Seraphine has said in a study of treatment effectiveness is that the effects of maturation. So maturation basically, it's like take it literally. See, human beings, they grow up and then they mature and then whatever. So when you're giving a drug, I think at the beginning stages of giving the drug, that's like the baby steps. And then eventually when you can measure whether the drug, upon the maturation of whatever experiment you're running and how long, maybe you say you're going to administer this drug over 8 weeks. So at 8 weeks, I'm assuming the experiment has reached its maturation, and then you're going to see whether the effects are what you expected them to be, and you can measure them. I don't know if I'm making sense. That's how I've understood it.
What I'm getting from the internet, maturation refers to the possibility that natural processes which occur over time within the study participants, such as growing older, hungrier, more fatigued, wiser, may create a false treatment effect or mask a real one. So these are things that are happening other than, like if you're using a certain study group and you're giving a drug, both the control and the others who have gotten the drug, they probably get hungry, they probably have some things that are beyond your control. That's the maturations. That's what, according to, like in terms with research, that's what maturation means.
Okay, then by that definition, then the answer still remains measured, yeah? Because you can still measure the effects of maturation then.
So someone is saying they think now the answer is eliminated, sorry. Perhaps patience, if you don't mind, written, so that we can see whether the drug is effective irregardless of age and maturation. That's an explanation for why elimination, so that we can see.
The reason I think the answer is elimination is, as someone was explaining, so if, okay, for example, we are testing a drug over 10 years. So we take 2 different groups so that even though they grow or mature within these 10 years, we can eliminate maturation in these people so that we can't, okay, because if we don't have a control group, we may end up saying that the drug is ineffective, but maybe it's because of the maturation. Okay, I don't know whether you're getting what I'm trying to say. We can eliminate maturation and maybe things like aging, because if some effects are the same across, it means it's an effect of maturation and not that the drug is ineffective. I hope you've understood what I'm trying to say.
We'll say kinda. Someone heard?
Some points with patient's answer, but I feel like in research we never really eliminate, so the answer would actually be reduced, because even might put in some measures to eliminate it, but you cannot eliminate something completely. Like you can reduce it though. So yeah, I feel like the answer should actually be reduced, and I'll share something in the group, then you can have a look. It's long, but you can have a look at it.
Excuse me. Excuse.
Yeah, go ahead.
According to this question, according to my understanding, it's like we are doing an example. We are doing experiment. We have a control group where we already having existing results which are already given as positive results, and we have another group that we want to do some research on. Now, the research on with the same objective with the ones that had given us positive results. Now we want to compare the two with one that had given us a positive result, acting as a control group. Now, the reason for including the control group is to help us know when we've reached our target, when we've reached our targeted result. Hence, the answer here, according to my understanding, will be measured. The control group will help us measure when we are already achieving our desired result. Me, I'll go with the measured.
Me too.
Measured, since most are going with measured, I think we can go with measured. Yeah, we can go with measured. And proceed on to the next. But you can just put a star so that once people are going through it.
Mr. Speaker, I'm saying we are dealing with two drugs. One is already existing, maybe in the market, it has been used even before. That is a standard drug. You're comparing it with the test drug, the one you are coming up with, the one you want to test against that. Now you are comparing the effectiveness of the already existing drug and the test drug. So whatever you're doing is you're measuring the effectiveness. No complication.
Perfect. I think yeah, you can agree now on the answer. But Sam, see you are
agreeing on the answer, but I think the first thing of getting the answer right is first by getting the definition or what the question is asking right. Because if we end up creating our own definitions for terms in the question, then we might end up with anything being the answer. I think that it's the best thing to get actually the actual definition and not what you think to be as the definition of what is being termed. Because I think maturation is actually a term used in research, and
someone told us, let us stay scientific. Yeah.
Because I'd actually agree with the definition of the other persons, as long as you show us that the definition you're using to describe maturation is actually the correct definition of maturation. So why are you guys getting that definition of maturation? So that you can actually correlate that to the answer that you're giving.
Google. Like that's what Janet said she got to answer.
Just to read out something on the same, sent on the group chat, that the advantage of including a control group in a study of treatment effectiveness is that it helps reduce the effects of maturation. Maturation refers to natural changes or developments that can occur in study participants over time, independent of the treatment being studied. By having a control group that does not receive the treatment, researchers can compare the changes observed in the treatment group with those in the control group. Any differences in outcome between the two groups are more likely to be attributed to the treatment rather than maturation. This control group allows researchers to better assess and isolate the true effects of the treatment, reducing the influence of maturation as a potential source of variability in the results. So you take two groups of the same age, one group is a control, and the drug effects are observed over 10 years. In these 10 years, maturation occurs in both groups and effects are observed. So based on the explanation from Dr. Hasna, it seems that the answer should be reduced. The effects of maturation can be reduced, not measured. The effects of maturation can be reduced. And as Dr. Sam has told us, let us stay scientific.
But the key word here, treatment of effectiveness.
So we proceed with this. Increasing the sample size.
I was saying. I was saying the key word there is the effectiveness of treatment. That's what we are looking at. Forget about maturation. Okay, that's my take anyway.
So we leave it with a star.
The keyword is the effects of maturation, not effectiveness.
So in any paper, if you see a star on a question, know that the answer was debated upon, and you'd need to do your own further research. Should it be repeated? It's your choice. It's your choice. Yeah. Choice number 94. Already seen choice A. Someone has said choice A, that increasing the sample size decreases sampling error. Are we all in agreement that increasing the sample size decreases the sampling error? Because based on the choices, of course, it has to be A or B. They are the two opposite choices. So it has to be A or B. Moving on to number 95. Interval or ratio data should be represented as a dash. Interval or ratio data should be represented as a histogram, bar graph, scatter plot, A and B. Interval or ratio data should be represented as a what?
I think D.
A and B. Someone else is saying they think A. Someone else B. Perhaps you can see on the group. Yeah, so based on what has been sent on the group, that a histogram can only be used with classified metric data such as interval and ratio data or data. So a histogram can only be used with classified metric data. This is interval and or ratio data. So that tells us histogram is the answer. Based on that. Moving on to number 96. Unless people have differing opinion. Number 96 question asks that the gender of patients is an example of what? A ratio scale, a nominal scale, an ordinal scale, or an interval scale. I will say that is an example of a nominal scale. That is an example of a nominal scale. That is an example of a nominal scale. Moving on to well, now on that, perhaps someone can just
Can
tell us.
we just define the types of scales, please?
Yeah, the ordinal scale, the interval scale, the ratio scale. In our dear researchers, Dr. Sonia, our published author, is you can tell us more on an ordinal scale, interval scale, ratio scale. In our ordinal scale, example or an example is levels of education. So we are having levels, level 1, level 2, level 3. Yeah.
Okay, the answer is nominal scale, because I can see example, a nominal scale with example. So gender, marital status, religion, race, hair color, country. They are all examples of nouns that do not require rank or order. So a nominal scale is a measurement scale in which numbers serve as tags or labels only to identify or classify an object. It deals with non-numeric variables where numbers have no value.
Ah. So that's nominal, ordinal.
Okay, yeah. So basically, nominal is the one that cannot be ordered from high to low, or they can't be ordered based on values. And then I'm seeing, is there an ordinal scale in the choices? An ordinal variable are those ones that can be ordered from high to low.
Great. So those levels of education. Then the other thing sent, interval data. This is the difference between measurements, but no true zero. Example includes temperature, standardized exam score. So difference between measurements, but no true zero. Yeah, no true zero. Then ratio data is the difference between measurements where true zero exists, such as height, age, weekly food spending. Then something else, an interval scale is one where there is order and the difference between two values is meaningful. Examples of variables include temperature and credit score. So that's an example of an interval scale, one where there is order and the difference between two values is meaningful. Chat box. The 4 levels of measurement: nominal, ordinal, interval, best measure, mean. Also there's best measure. Okay, you consider those 4 levels of measurement of data, but just the other thing is that nominal and ordinal are qualitative or categorical. That's another term. Nominal and ordinal are qualitative or categorical, whereas interval and ratio are quantitative or numerical. So nominal and ordinal, qualitative, interval and ratio are quantitative. Moving on to number 97. The effects of ibuprofen on the cardiovascular system is salt and water retention, bradycardia, vasoconstriction, tachycardia. So here, I think we've discussed this before, and we saw NSAIDs do cause salt and water retention. Such, they need to be given with caution. Patients with heart failure, with hypertension. So salt and water retention. Number 98, salbutamol induces bronchodilation through stimulation of water
Is that a
receptors.
cardiovascular effect? Is that a cardiovascular effect? Salt and water retention.
Salt and water retention. Let's just go back. What are your thoughts? What are your thoughts? There's bradycardia, tachycardia. I think the other choice was. Seraphine can just scroll up. see yeah, or vasoconstriction. But on salt and water retention, that is a cardio effect. Salt and water retention, yeah, even on the vascular, right? When you retain more water, there's more pressure on the vascular system. There's more, the afterload is also increased. So hence, my thoughts are that is a cardiovascular effect. But what are your thoughts on the same? Or which among the choices, which one would you go for?
I will go for vasoconstriction.
How?
I think they cause vasodilation.
Yeah, because yesterday we said they're associated with. No, wait. Can you explain how does ibuprofen or NSAIDs cause vasoconstriction? And among the given choices,
They inhibit prostaglandin synthesis, so meaning there will be no PGE and prostacyclin, which are vasodilator. So meaning they will lead to vasoconstriction.
But is it a major thing as compared to sodium and water retention? Like if we were to choose the best answer, the something that has been shared on the group about the same. Is sodium and water retention isn't, are you disagreeing that that is not related to the cardiovascular system?
Because I can read sodium and water retention, hypertension. Okay, it also causes affect arterial vasodilation. That's not even in the choices though. So it's only salt and water retention.
Yeah. Well, personally, I think that's the best answer. Maybe if someone
wait.
Uh-huh.
That was the effect on the kidney. Cardiovascular, I'm seeing actually. Vasoconstriction.
Sam?
Yes, could you describe your process of salt and water retention? When you want to pay vasoconstriction, but we describe kwanza salt and water retention, how it comes about.
Okay, what I know is through that inhibition of, okay, first things first, let me say first things first, is that when we are talking about NSAIDs and anything to do with the cardiovascular system, we always, the big thing is always about sodium and water retention. When we go essentially to the renal system, that is when we mention on the them reducing vasodilation of the afferent artery. But now on the mechanism of sodium and water retention is that PGE2, we know it acts to vasodilate the afferent arteriole. And we know when the afferent arteriole is vasodilated and it has a wide lumen diameter and the efferent arteriole has a narrow lumen diameter, there's more filtration taking place, so you lose more sodium, you lose more water. But if you inhibit the COX enzyme, meaning the vasodilation is not present, meaning the difference between the lumen radius is not that big, hence there is less sodium and water filtration into the renal tubule, leading to more sodium and water retention. So that's my understanding of how they cause sodium and water retention, leading to hypertension leading to worsening of heart failure. Yeah.
I think that is
According to
agree with that answer.
explanation, salt and water retention is more of the kidney. And when you come to cardio means heart and vascular means the vessel. So like what does need the ibuprofen do to the vessel and the heart? Does it constrict them? Does it dilate them? I don't know. But I would have gone for vasoconstriction. Yeah.
Some.
Salt and cardiovascular system, cause cardiologist, because basically when they give loop diuretics, because basically that has an effect on the cardiovascular system, basically, because if it didn't have an effect on the cardiovascular system, then it will change. And basically when there's a lot of water retention in the body, that affects the cardiovascular system, since we're basically having, it actually leads to the hypertension and the heart rate increase. I mean, the workload of the heart being overstretched. So it's actually cardiovascular effect.
Let me share something here. Let me just share a screenshot. Yeah, that there's an expert here on the Queen's language saying that, guys, not to throw a spanner in the works, but B and D are juxtaposed. They can't be both correct or both wrong. Then prostaglandins have a role in increasing sodium and water excretion. NSAIDs cause retention. Induction of nephrotoxicity due to NSAIDs is via vasoconstriction, explaining the vasodilatory role of prostaglandins. Do describe which of these is cardiovascular or renal is a problem. Do describe which of these is cardiovascular to renal is a problem, or renal to a problem. I don't know why. but let me say something about NSAIDs in general, according to the book of Basic and Clinical Pharmacology. Because as much as they can cause a vasoconstriction, again, this is my 2 cents. The major thing with NSAIDs is about fluid and water retention. That is the major, fluid and sodium retention. That is the major thing we were always mentioning with NSAIDs. Yeah, because if we say that that is on the renal, the sodium and fluid retention is on the renal, then will we now say that furosemide is used in heart failure, but it doesn't interact with the cardiovascular system? You know, I don't know. By furosemide reducing or excreting more fluid, we say that is a plus on the heart, because fluid has to do with afterload. But let me share this. I'm just thinking out loud. Anyone can interject on the same? And blood pressure, ibuprofen can cause a temporary increase in blood pressure. This if it is more pronounced. Okay, risk of cardiovascular. Some studies suggest that it may be associated with heart attacks and strokes. The other thing, fluid retention. They can cause fluid retention, which can increase the workload, platelet function. They can interfere. While this effect is temporary, should be considered in the okay. I guess so that we can move. A star can be added to the same question. so that at the end of the day, someone can go with the right or the
Guys.
choice the best.
Okay, so my thinking is the person who thinks that salt and water retention is not cardiovascular, it's just because it's that the kidney is being involved. But remember, when regulation of hypertension, you're talking about the RAS system. And the RAS system is like big on the control of cardiovascular effects, right? That's where the long-term pressure control is. So water and salt retention will affect in that. It will increase hypertension, and that's it's not like a short outcome. So it's a long outcome. So like, you get what I mean? I don't know if
Yeah. I get what you mean.
Yeah. It just because it has an effect on RAS doesn't mean it's the kidney, it's the cardiovascular system, because RAS is what controls the blood pressure of the cardiovascular system. So if there's salt and water retention, that means it's a problem for hypertension and for other heart diseases such as heart failure, because you already have salt and water retention in heart failure, and in hypertension the blood is already increased. For vasoconstriction is not like the reliable answer here, because we have said COX-1 vasoconstriction, COX-2 vasodilation. So it depends on the different NSAIDs, although we have a specific example here. And yeah, so that's what, I don't know if that makes it easy for you.
Okay. And also, this is just a curiosity. This is actually AI that is writing all this. It's not someone calling themselves AI. I didn't know. Technology. Anyway. So let us, again, I will ask you, let us not throw spanners into the works. I've learned that today. I'll use it a lot. So let us not throw spanners. I think it's spanner. Yeah. Moving on. So salbutamol, I think it's a beta 2 agonist. There we can all agree and fly, because we've spent a lot of time, right? So unless someone has something to add on salbutamol. Okay. The cardiovascular effects of antihistamines include all of the following except cardiovascular effects of antihistamines include all of the following except tachycardia, hypertension, cardiac arrest, and bradycardia. So the cardiovascular effects of antihistamines will include all of the following listed there except so again, on cardiovascular effects, now here we are looking at the antihistamines. What's the effect of antihistamines on our cardiovascular system? Do antihistamines, histamine itself causes tachycardia. So can an antihistamine cause tachycardia? I mean, just thinking out loud. Histamine will cause tachycardia because of the vasodilatory activity leading to the heart pumping more, Personally, I'm thinking just that antihistamines do not cause tachycardia, but I'm not sure. I'm not sure. In the chat box, I'm seeing first generation antihistamines. Oh yeah, first generation antihistamines have anticholinergic side effects. So if they have anticholinergic side effects, they'll be associated with tachycardia. Will be there, right? Tachycardia is an anticholinergic. Hypertension, cardiac arrest, bradycardia, prolonged QT intervals.
If tachycardia is there, also bradycardia is there. Because anticholinergics, tachycardia is followed by bradycardia.
So side effects as shared on the WhatsApp group, side effects of antihistamines, we are seeing on the cardiovascular specifically, they cause a dose-dependent sinus tachycardia, reflex tachycardia, atrial refractory period prolongation, and supraventricular arrhythmias. Second generation, that side effect is not there. Severe CNS and cardiac side effects on the CNS, learning, memory, headache, confusion, hallucination. So based on that, the bradycardia, I think we go with now the circle answer, yeah. Yeah. Yeah.
How cardiac arrest? Explain on cardiac arrest. How does it cause?
On cardiac arrest, you know, now we decided not to throw a hammer onto the wax, so we said it's either bradycardia or tachycardia.
Okay, I was saying, you said first generation antihistamines, they have anticholinergic effect, right?
Yeah.
Okay, examples of anticholinergic, like buscopan, is it one of them? Or I'm just confused.
Buscopan, okay, it does have some anticholinergic activity. Go ahead.
When you administer buscopan, at first there is tachycardia followed by bradycardia. So that's why I was telling you, if bradycardia is there, then tachycardia is also there. If we take it in that line, I'm not sure. I'm just asking.
Yes, so antihistamine experts, what were Piriton, tell us how do you feel? Have you ever gotten a cardiac arrest? I'm a hypertension, like yeah, it's short antihistamines generally. If we're asking for explanation of the side effects of antihistamines. Saying that something on chat on the WhatsApp group is that the H2 receptor antagonist can cause bradycardia, but that is H2. H2. See, right now we are interested in the H1, but this lyrics in 3 recently the third associated. I think H2, that is the likes of ranitidine and their related brothers. Again, on this was to save time. We'll just go with the bradycardia to be bradycardia.
I have a question.
Yeah.
But not really related to not to as according to the question we have up far. Now, when managing allergic reaction, muscle type 1 in hypertensive patient, what is the drug of choice? Because it seems antihistamine will increase the hypertension.
So question is, when managing an allergic reaction type 1, but now for type 1, given it's an anaphylactic shock, we'll just have to give that adrenaline. Then in the case of these cardiovascular effects that we are seeing here with the second generation antihistamines, we do not see the second generation antihistamine playing any role on the cardiovascular. So I guess we can give that adrenaline for the acute management. And if you have to give an antihistamine, then a second generation antihistamine such as loratadine would suffice. We don't have to give the first gen, because they are the ones that are associated with all these side effects. Have I answered them?
Thank you.
Welcome. Moving on to number 80. Which one of the following is not an effect of digitalis? We did digitalis yesterday. I'm expecting people to be shouting on the chat box. Yeah. Which one is not an effect of digitalis? Is it thrombocytopenia, anorexia and nausea, decreased concentration of LH and testosterone, or decreased serum concentration of FSH and estrogen? Not an effect of digitalis. Right? Study we did cardiac glycosides. Not an effect of digitalis. Yeah. I'd go with choice A. Yeah. So is a big fan of the cardiac glycoside. So Mystique is also saying choice A. So his answer saying that digital does not decrease serum of FSH and estrogen, but they're agreeing that digitalis does decrease concentration of LH and testosterone. Digital is here, so it's thrombocytopenia. Yeah. Moving on to the next question, number 11, talking about ACE inhibitors are dash teratogenic, cause productive cough, can precipitate hypertension, must be avoided in nephropathy. mistake again is saying choice A, that ACE inhibitors are teratogenic. They are not to be administered during pregnancy. So in terms of the RAS system, ACE inhibitors, ARBs, we again looked at them quite in detail. So we can just continue to the next question. Number 102, the respiratory reactions to sulfonamides include all of the following except. The respiratory reactions to sulfonamides include all of the following except migratory pulmonary infiltrates, chronic pneumonia, asthma, or bronchodilation. The respiratory reactions to sulfonamides include all of the following except. So sulfonamides. Seraphine, can you scroll just bit down? Yeah, so we'll finish with that number 90, not 90. Just now the previous page. Yeah, I'm seeing the energy levels have gone down, just below the quinine choice. I think there's a question in that on that page after quinine. Yeah. So we'll finish with that. Yeah. So meanwhile, you can go to sulfonamides. Respiratory reactions to sulfonamides except asthma, bronchodilation, chronic pneumonia, or migratory pulmonary infiltrates. So yeah, these are the last 3 questions. It's time to make your mark. Yeah, I'm talking to you. Let's see. Lucy, it's time to make your mark. Respiratory.
So would be D? Would answer D.
D.
Because yeah, because they cause difficulty breathing, and I'm assuming it's because they will cause bronchoconstriction, not dilation.
Okay. They cause bronchoconstriction, not dilation. And so the answer is D. Moving on to number 1. I feel like it's always been 103. Quinine can cause tinnitus. Answer has already said tinnitus, a ringing in the ears. Yeah, quinine can cause, tinnitus. What are the other side effects of quinine? Was it called synchronism or something? Hypoglycemia, tinnitus. What else was there? The side effects of these quinines: bitter taste. Yeah.
Dryness of mouth.
Dryness of mouth. Side effects of quinines in general, or the common ones. I've said hypoglycemia, tinnitus, dryness of mouth.
There is black water fever due to hemolysis RBC.
Nice. Black water fever due to hemolysis of red blood cells. Yeah. Wait, what's the presentation in black water fever? You know, we may be saying black water fever, and what's the presentation? Just the general. How would you say this is black water fever?
I'm not sure. I'm thinking. Is it dark urine? I don't know. I don't know, honestly.
Yeah, correct.
Dark urine.
Oh, thank you.
Nice. Yeah. Remember, this stuff has that oxidative potential, so yeah, so it can go to, it can cause that hemolytic anemia. Yeah. Any other side effects of quinines, perhaps, that we may have left out, and you feel this one has to be said?
Not a side effect. Just to say, quinine is usually given with dextrose just to avoid the hypoglycemic effect.
Nice. Quinine is usually given dextrose to counter that hypoglycemia, right? Don't just say, it causes hypoglycemia. Then you just give, right? Anything else about quinines in general? Or let me ask, what's the mechanism of action of these drugs? We have one last question to go. Mechanism of action of quinine or of chloroquine. Let me say chloroquine specifically.
So
Mechanism.
for the malaria parasite, it is the conversion of hemoglobin to heme and then hemozoin. So it converts heme to hemozoin, right? So hemozoin is not toxic to Plasmodium per se. So the drugs will prevent the conversion of heme to hemozoin so that heme is high, and that heme is what is toxic to Plasmodium.
Nice. So yeah, the drugs will come in to inhibit that conversion of heme to hemozoin. Then heme is very reactive to do its thing, causing lysis of the parasite and of the RBC. Yeah, nice. Yeah, so I think that is, yeah, that's side effects. I think we've mentioned them. I'm just looking through the slides to see if there's any particular one that we may have left out. Oh wow, there's this really unique one here, retinopathy. Yeah, retinopathy. Yeah, remember these drugs can cause chloroquine can cause blindness. Yeah, retinopathy, ototoxicity. Yeah. There's a person in COVID when this hydroxychloroquine was rumored to be the cure. They were given a high dose and led to blindness. Moving on to the last question, which dextrose strength has been answered. Perfect. Uses of quinine, side effects, nausea, vomiting, diarrhea. That's a whole pharmacist telling us nausea, vomiting, diarrhea. Finally, on the last question, all the following drugs can cause a neurological problems, except all of the following drugs can cause a neurological problems, except, and kindly clarify on which dextrose is given. Is it D5 or D10? Is that question? Yeah, on when
5%.
5%. So D5. Say D5. Yeah. But remember, in our setting, when resources are limiting, you go with whatever is there. If there's no D5, just give that D10. So all of the following drugs can cause neurological problems, except neurological problems, except. So these are the first line anti-tubercular drugs. So which one does not cause a neurological problem? Isoniazid, ethambutol, fampicillin, or pyrazinamide. So which one does not or is not associated with causing any neurological problem?
Parazinamide.
Local clown is saying parazinamide. Are we in agreement? Pyrazinamide. And meanwhile, you can tell us, no, that's not significant here. So are we in agreement? Pyrazinamide?
Google is saying rifampicin.
Yeah, also rifampicin doesn't have any. I don't know if parazinamide has some, but I don't know of any as well. Because ethambutol does have some neuritis for the optical and peripheral. Isoniazid pair.
So
we're left with rifampicin
and pyrazine. So we've eliminated isoniazid and ethambutol. So rifampicin or pyrazinamide. Rifampicin or pyrazinamide. Someone is agreeing with you that pyrazinamide is not associated with any neurological issues. So pyrazinamide seems to be the consensus that pyrazinamide is great. So we discussed TB before in detail and do so again. So that brings us to the end of the discussion. Thank you for being an active audience. Yeah, there was never a dumb comment.
You guys.
Yeah.
Unaweza enda ile swali ilikuwa nauliza ADR za nini za digitali.
So ADRs of digital is so I think to scroll back to that question. Uko na any
sawa.
So thank you. See you all in 3 hours. Hope you enjoy your lunch. Yeah, tupatane afternoon. At 4.